you, Laura. So hello, everyone. So today I'm gonna begin with a few basics regarding genetic tests, and then we see that how diverse are our genomes, and we see that in some cases, the clinical signs in PCD depend on the gene that has been implicated. So I guess that after Suzanne's talk, you know that we have cilia in our airways, but also in our uterine tubes in women and in the flagella of our sperm cells. So if you cut those structures, you can observe what you see on the right, which is the axoneme, which is made of 9 outer doublets and a central pair in the middle. The motility of the cilia is done through the outer dining arms in blue and the pink dining arms, inner dining arms, that are the molecular motor of our cilia. There are more than 50 genes that have been implicated in PCD. Most of them encode for structure of the cytoskeleton of the cilia. The most frequent one encode for genes that are proteins, mainly coding for the outer dining arms, like DNAH5 or DNAH11. PCD is a recessive disease, which means that we have all of our genes most of our genes in 2 copies. And to get a recessive disease, we need to have mutation in both copies of the gene. So here on the left, you have a patient with 2 different pathogenic variations. And one is inherited from the father and the other one from the mother. And both parents do not have PCD. So that's the definition of a recessive disease. So unfortunately, most of the sequencing techniques are not able to make the difference between both variations, between on 2 different copies, or the 2 variations being located within the same copy. So to make the difference, we need to analyze the DNA of both parents. And in some instances, both variations can be located in 1 copy that is inherited from only one of the parents. So in the problem, there's 1 mutation that is probably missing, or maybe that's another gene that's implicated. So that's why we always request samples from the parents. So what's the structure of a gene? It's made of exons, represented here by the red boxes, that encompass most of the coding regions. That the coding regions are the region that encode for the proteins that make our organisms. The exons only represent 1.5% of our genome, but they host most of the pathogenic variants. However, some mutations can lie in between the exons or in regulatory regions. So most of the sequencing techniques target only those exons. So how do we get from the gene to the proteins? So let's start with a genomic DNA with the example of a gene with 6 exons here in red. Then we get to the RNA by removing the intervening sequences between exons. And we get the RNA, which is the type of molecules that may be a future options for potential treatments for PCD. So once you get the RNA, it's going to be read by the cell from the beginning to the stop, adding amino acid 1 by 1. The amino acids are the bits that make the proteins. So in the end, after what we name translation, we get the full protein corresponding to a given gene. So our genomes are very diverse. So for example, if you take on the street 2 different people, they're going to have about 0.4% of differences between their genomes. So sequencing has been improved a lot by the next generation sequencing techniques that enable to sequence a lot of amount of DNA in just one experiment. That's a word that covers a broad range of different techniques. For example, in a patient, you can perform a targeted gene panel. We can gather only the 50 or 53 genes that are responsible for PCD. Or you can perform whole exome sequencing that sequence all the exons of all or 20,000 genes. Or you can, in rare instances, perform whole genome sequences that sequences everything, exons, and also regulatory regions. So as you can see, with a targeted panel, you're gonna highlight several hundreds of variations, being together benign and pathogenic variants. So that's a lot. It's even worse if you perform whole exome sequencing, because you're gonna highlight for each individual about 30,000 variations. And with a whole genome, your output file will be about 5 million variations. So as you understand, the problem is not anymore to sequence. The problem is to understand what is benign and what is pathogenic. So for example, if I sequence my own exome, I will find 30,000 variations, being benign or pathogenic. And after sorting all those variants, I'm probably a carrier for something between 10 and 100 pathogenic variations. I may not develop a disease because those variants are located in genes mainly implicated in recessive disease, and I'm just a carrier. So all around the world, the labs classify the variations in 5 main groups. Pathogenic and probably pathogenic variations have to be taken into account for genetic counseling. On the other hand, the variations of uncertain significance, or VUS, the probably benign and benign variations shouldn't be taken into account for genetic counseling. So the problem are those VUS, because we're not able to tell to the patient that it's pathogenic or benign. So as you guess, it's all not always that simple, just finding right away the pathogenic variants in the right gene, just out of a desert of genomic data. In many instances, we're going to find in the patient one likely pathogenic variation, but only in one of the 2 copies. He or she will also carry one heterozygous VUS in another PCD gene, and maybe one heterozygous variation in a gene, a candidate genes you don't know the function of. So maybe I'm going to try to remove the, if I manage to. Because I cannot remove the pointer so that you can see the right of my, better see the right of my screen. Right. So there are some types of mutations that are pretty obviously pathogenic. These are the one in orange on the top. So they lead to a premature stop codon, which is the signal that tells the cell it should stop producing the protein. So it's upstream the usual one. So the cell is going to detect that it's not normal. It's going to degrade the RNA and there won't be any protein. So we know that those variants are usually pathogenic. It's a bit more complicated for missense variations that only replace one of the beads, one of the amino acid within a very large protein by another bead, another amino acid. So it's quite tricky usually to determine if it's pathogenic or not. So how can we decide if a missense variant is benign or pathogenic? So we can rely on very large databases, gathering genomic data from thousands of people from the general population. And if we find that the variant is very frequent in this database, it cannot be pathogenic. We can, of course, have a closer look at the type of change and to look especially where it's located within the protein. A very powerful tool is to look at conservation through evolution. If the amino acid is conserved from very simple organisms to humans, then probably we shouldn't change this bead within the protein. Another way to decide if it's pathogenic or not is to perform a family study and looking for the variant, for example, in a relative that also has PCD, and if he or she doesn't carry the variant, then we exclude the gene and we have to go on with the genetic investigations. So I'm going to give you the example of a variant we've identified in a patient in the NH5. This means that an arginine is replaced by a glutamine in this individual. So we go and have a look at this large population database, and we see that 25 people who do not have PCD have this variation in both of the copies of the NH5. In addition, it corresponds also to a 7.5 heterozygous carrier proportion. So this together makes these variants much more too frequent to be responsible for such a rare disease like PCD. So that's rather in favor of a benign variance. Again, for these variants, we can look at conservation in vertebrates, and you can see that you have a glutamine that corresponds to the variation in two vertebrates, which are the dolphin and the killer whale. So in the proteins of those vertebrates, there's a glutamine, and this protein is able to work properly. So that's, again, another element that make us classify this variant as likely benign. So the clinical signs in PCD can sometimes depend on the gene implicated. So in PCD, in some cases, the gene implicated encodes for outer or inner dining arms. So we know that we have cilia in our airways, but we also have cilia in a structure in the embryo that is named the nodal cilia, and that is responsible for the asymmetric lateralization of our thorax and abdomen. Those cilia are the reason why we have the heart on the left and the liver on the right. So when patients have mutations in those dining arms, the nodal cilia have motility defects, and then the patient will have a situs inversus randomly in half of the cases. On the other hand, if we carry a mutation in a gene that encodes for a central structure that only exists in airway cilia but does not exist in nodal cilia, then those nodal cilia will have a normal motility, and the patient will not have any lateralization defects. Very recently, it has been shown that some people who only have a situs inversus without any airway disease carry mutations in genes that encode for proteins located within the outer tubules. There are 5 genes now known so far. So people will only have a heart on the right, and they won't have any lung disease. So we used to hear that people with PCD were infertile, and we know now that it's not always the case. So in 2000, 2017, we've done a study in about 90 adult patients with PCD with fertility data, and we've found that some people with mutations in DNH5 or DNH11 had babies without the use of IVF. So we wondered why, and we tried to explore this issue. And a part of the answer came from the implication of the DNH17 in male patients who only have isolated male infertility and no lung disease. So we've shown that, in fact, this protein, together with another one, are the autodyning arms only in sperm cells in the flagella, but they're absent from airway cilia. And conversely, DNH5 and DNH11 are, of course, in the airway cilia, but absent from the sperm cells, which may explain why some male patients had babies without the use of IVF. So my take-home message would be that there are 30,000 variations in an exome, that we all carry pathogenic variations between 10 and 100, even if we do not have a genetic disease, and that in PCD, clinical signs and ciliary defects depend on the gene that has been implicated. So I thank you, and I thank our team working on diagnostic and research in PCD, Rana Mitri for the electron microscopy data, Bruno Louis for the video microscopy studies, and the ADCP French advocacy group.